Okay, so good afternoon, everyone. My name is Anjmani, and I'm joined alongside our group members.
Hi, my name's Cooper.
And my name's Elika.
I'm Luna.
And I'm Isabella.
All right, so we'll move over to the table of contents slide. And basically, throughout our entire presentation, we'll be covering a variety of different topics. So we'll go through the introduction, we'll cover pathogenesis, we'll cover molecular mechanisms, and then, of course, we'll also cover different statistics and distributions that we felt were important to include. And then, of course, we'll also include our discussion and conclusion. Next slide. So for the introduction, basically we're going to cover what is tobacco and how it is related to cancer. Next slide. So first, what is tobacco? Before we talk about how tobacco causes cancer, we felt that it was important to understand what tobacco actually is. So tobacco is essentially a type of plant where the leaves have been dried and then processed to make things such as cigarettes, cigars, pipe tobacco, hookah, and snuff, for example. In more recent years, with the development of technology, we've noticed a rise of nicotine, which is derived from the tobacco plant and is present in a lot of electronic cigarettes and vaping tools. But why does the tobacco cause cancer? So while nicotine is the substance that promotes the actual addiction itself, it's not the primary cause of cancer. The tobacco smoke is where the main cause of concern comes in. Each time a cigarette burns, a mixture of almost 7,000 chemicals is produced. And at least 70 of these substances have been found by scientists to be carcinogenic, which means they are known to cause cancer in people. These carcinogens then enter the body through the lungs, but they don't just stay there. Many of them enter the bloodstream and travel to various organs throughout the body after they've been inhaled. So because of this, the smoking increases the chances of different types of cancers like lung cancer, as well as cancer of the mouth, throat, larynx, esophagus, stomach, just to name a few places, and even acute myeloid leukemia. In addition, tobacco causes cancer because the carcinogens damage the DNA, which of course we know the DNA contains the genetic instructions that control how our cells grow and divide. And if our cells can't properly grow and divide, then that's when the cancer starts to form. Typically, DNA damage can be repaired by the cells, but with regular exposure to tobacco smoke, the damage occurs quicker than it can actually get to heal itself. So these DNA damages build up over time and result in mutations that let the cells grow uncontrollably and ultimately lead to cancer. So why does any of this matter anyway? So around the world, tobacco continues to be the primary preventable cause of cancer and cancer-related deaths. So if we understand how tobacco can contribute to cancer, we can enhance public health knowledge since it's a behavior that can be stopped and changed if people are aware of what the effects are and how they can affect the body and health in general. So throughout our presentation, our group will discuss how tobacco exposure causes cancer, the molecular mechanisms underlying DNA damage and mutations, the most important demographics that are impacted, and we'll also cover two studies that we felt were important and improved our knowledge of tobacco-related cancers. In addition, we'll also talk about what prevention methods there are, and why giving up smoking is still one of the best methods to lower your risk of developing any sort of cancers to begin with. Next slide.
Okay, hi, my name is Luna, and I will be speaking about pathogenesis. The question I will be answering today is, how does tobacco exposure lead to cancer in the body? Next slide. First, we have to ask, what is pathogenesis? Pathogenesis is generally known as the biological process by which a disease develops. Specifically, I will be speaking on pathogenesis in relation to how tobacco causes the disease we know as cancer. This process occurs as soon as tobacco smoke enters the body. As stated before, tobacco smoke contains 70 known carcinogens and thousands of additional chemicals. Although nicotine is addictive, carcinogens is what is responsible for cancer. And for future reference, the definition of carcinogens is anything capable of causing cancer in living tissue. Next slide. Here I have a diagram of how tobacco moves through the body. So first, the chemicals from the tobacco smoke enters the lungs, then it is absorbed into the bloodstream, and lastly, it reaches organs throughout the body, such as the pancreas, kidneys, and bladder, to name a few. As we can see, tobacco doesn't just stay in the lungs. Carcinogens circulate throughout the body, which explains why smoking is linked to cancers in many different organs. Next slide. I will now explain how DNA damage is caused by tobacco carcinogens. There are two major tobacco carcinogens, are polycyclic aromatic hydrocarbons, also known as PAHs, and tobacco-specific nitrosamines, TSNAs. When these carcinogens enter the cells in our body, metabolic enzymes convert them into highly reactive compounds. These compounds cling to the DNA and form DNA adducts. DNA adducts deform the DNA molecule and inhibit DNA from replicating properly. If the DNA is not fixed before the cell divides, permanent mutations happen. Over time, these mutations accumulate and increase the chances that a normal cell will become cancerous. And I just put how it flows. So it goes from tobacco smoke to the PHHs and TSNAs, the metabolic activation, then to DNA adducts, and then DNA mutations. Next slide. This slide is about DNA repair failure and uncontrolled cell growth. The defense systems in our cells frequently stop cancer from spreading. Enzymes that repair DNA constantly check for damage and fix mutations that it encounters. A protein called P53 can pause cell division and initiate programmed cell death, also known as apoptosis, if the damage is too much to reverse. But over many years, tobacco smoke repeatedly damages DNA. When mutations affect genes that regulate cell growth, such as the tumor suppressor gene, TP53, damaged cells continue dividing. One of the characteristics of cancer is this uncontrolled growth. Next slide. Next, I will explain clonal expansion and tumor formation. When genes that control cell development are mutated, the damaged cells start to divide uncontrollably. It lives and transfers its mutations to all of its offspring cells rather than undergoing apoptosis. Because several genetically identical abnormal cells are created from a single initial mutant cell, this process is known as clonal expansion. These mutated cells develop more genetic modifications when they divide, making them even more abnormal. Certain mutations aid cells to prevent apoptosis, while others make it possible to disregard signals that often halt cell division. These benefits help abnormal cells to reproduce and persist significantly more than successfully than normal cells. The number of cells eventually needs more nutrients and oxygen than diffusion can supply. In order to sustain its development, cancer cells promote angiogenesis, the creation of new blood vessels that provide nutrients and oxygen to the growing tumor. This growing mass of cells eventually turns into a cancerous tumor, as I show in the picture below. Compared to benign tumors, malignant tumors can eventually spread or metastasize to other areas of the body by invading surrounding tissues. At this point, a normal cell has developed into cancer due to years of repetitive DNA damage by tobacco carcinogens. Next slide. In this slide, I will talk about why tobacco causes cancer in so many different organs. Although lung cancer is frequently linked to smoking, tobacco smoke contains toxins that impact almost every organ in the body. The carcinogens goes through the lungs, alveoli. Alveoli are small air sacs that allow oxygen to enter the blood. The alveoli have a very thin membrane that are covered by a thick network of capillaries that allows the chemicals to reach the blood quickly. Carcinogens quickly diffuse into the bloodstream and they are carried throughout the body. The lungs experience the greatest exposure because they come into direct contact with tobacco smoke. Lung cells are repeatedly exposed to carcinogens such as PAHs and TSNAs, and these chemicals form DNA adducts, leading to mutations that can eventually result in lung cancer. Next, I will be talking about mouth and oral cavity. Every time someone breathes in cigarette smoke, their mouth and oral cavity are almost immediately exposed. Carcinogens frequently cause damage to the lining of the mouth, the tongue, the gums, the throat, raising the risk of cancers of the lips, the tongue, the oral cavity, and the larynx. And then the kidneys are also affected. Toxins and waste products, including tobacco carcinogens, are continually filtered out of the blood by the kidneys. Kidney cells are frequently exposed to these chemicals during the filtering process, which raises the risk of DNA mutations that might result in kidney cancer. The bladder is also one of the organs most impacted. Because many tobacco carcinogens are eliminated through urine, the bladder is particularly sensitive. These carcinogens stay in extended contact with the bladder lining when urine accumulates in the bladder. And bladder cancer is closely associated with cigarette smoking, second only to lung cancer, because this frequent exposure destroys the DNA of bladder cells. And lastly, I will be talking about the pancreas. Instead of coming into direct touch with the smoke, the pancreas is exposed through blood circulation. DNA mutations, persistent inflammation, and abnormal pancreatic cell function can all be caused by tobacco carcinogens. When combined, these alterations greatly raise the chance of pancreatic cancer, which is among the deadliest types of cancer since it is often discovered at an advanced stage. Although each organ is exposed in a different way, the underlying process is the same. Tobacco carcinogens reach the cells, they damage the DNA, overwhelm the body's repair mechanisms, and allow mutations to accumulate over time. This is why smoking is not just the cause of lung cancer, it is a systemic carcinogen that increases the risk of cancer throughout the entire body. Next slide.
Now that we've talked about how tobacco enters the body and begins the disease process, I'm going to explain what actually happens inside our cells that eventually leads to cancer. There are 4 main mechanisms I'll be talking about today: DNA damage, mutations, oxidative stress, and how these changes build up over time to eventually form cancer. Next slide. The first step is DNA damage. Tobacco smoke contains more than 70 carcinogens, meaning they have the ability to cause cancer. On this slide, I've listed some of the most important ones. Every time someone smokes, these chemicals travel into the lungs and quickly enter the bloodstream. From there, they spread throughout the body and reach many different organs. However, these chemicals don't immediately damage DNA. First, our cells try to break them down using enzymes. Normally, these enzymes help remove harmful substances from the body. But with tobacco carcinogens, something different happens. Instead of making them harmless, the enzymes actually convert many of these chemicals into highly reactive compounds. These reactive compounds are unstable, so they begin interacting with important parts of the cell, especially the DNA. This is really the beginning of how tobacco starts the cancer process. Next slide. This slide explains one of the most important concepts in tobacco-related cancer, which is DNA adduct formation. The enzymes involved are called cytochrome P450 enzymes, especially CYP2A6 and CYP2A13. Their job is to metabolize or process tobacco carcinogens. During this process, they create highly reactive intermediates. These intermediates attach directly to DNA. When a chemical becomes attached to DNA like this, it's called a DNA adduct. You can think of a DNA adduct like a stain or a piece of glue stuck onto the DNA strand. Normally, DNA needs to be copied exactly every time a cell divides. But if a DNA adduct is sitting on the DNA, the cell's copying machinery can misread the genetic code. Instead of inserting the correct DNA base, it inserts the wrong one. At first, the body tries to repair this damage, but with repeated smoking, DNA adducts are constantly being formed. Eventually, some of them escape repair. Once that damaged DNA is copied, the mistake becomes permanent. That permanent change is called a mutation. So the sequence is really tobacco carcinogens are activated by enzymes. They form DNA adducts. DNA is copied incorrectly, mutations develop, and over time, those mutations can lead to cancer. Next slide. Among all the carcinogens found in tobacco smoke, 2 of the most important are called NNN and NNK. NNN is most strongly associated with cancers of the mouth and esophagus. After it enters the body, it's converted into unstable compounds that easily bind to DNA and create DNA adducts, which increases the chance of permanent mutations. NNK is slightly different. It's much more strongly linked to lung cancer. Inside the body, NNK is converted into another carcinogen called NNAL. Studies have shown that lung cancer is especially good at activating NNK, which is one reason why lung cancer is so common among smokers. So while tobacco smoke contains many harmful chemicals, these 2 compounds play a major role in damaging DNA and increasing cancer risk. Next slide. Once DNA damage occurs, the question is whether the cell can fix it. Normally, our cells are constantly repairing DNA. Every day, our DNA experiences damage from things like sunlight, pollution, and normal metabolism. Fortunately, we have repair systems that find mistakes and fix them before they become permanent. The problem with smoking is that the amount of damage becomes overwhelming. New DNA adducts are constantly being created with every cigarette. Some are repaired, but many remain. When the cell eventually divides, it copies that damaged DNA. If the damaged section is copied incorrectly, the mutation becomes permanent and is passed on to every daughter cell produced afterward. This is why smoking-related cancers usually don't develop overnight. Mutations slowly build up over years, sometimes even decades. The more damage that accumulates, the greater the chance that important genes controlling cell growth will eventually become affected. Next slide. This brings us to some of the genes that are commonly damaged by smoking. One of the most important is TP53, often called the guardian of the genome. Normally, TP53 acts like a quality control manager for the cell. It checks DNA for damage, pauses cell division so repairs can happen, or if the damage is too severe, tells the cell to destroy itself through apoptosis. But if TP53 becomes mutated by tobacco-related DNA damage, it can no longer perform those jobs. Damaged cells that should have died instead continue dividing and passing their mutations to new cells. Another important gene is Cras. Under normal conditions, Cras helps regulate cell growth and tells cells when it's appropriate to divide. However, smoking-related mutations can permanently switch Cras on. That means the cell keeps receiving signals to grow and divide even when it shouldn't. Other genes shown here like EGFR, RB1, PTEN, and CDKN2A can also be affected. Together, mutations in these genes remove many of the normal safety systems that keep cells healthy and prevent tumors from forming. Next slide. Besides directly damaging DNA, tobacco smoke also causes something called oxidative stress. During smoking, large amounts of reactive oxygen species, or ROS, are produced. These are highly unstable molecules that react with almost anything nearby. ROS can damage DNA, but they also damage proteins, cell membranes, and other important structures in the cell. This creates even more opportunities for mutations to occur. A study I used for this research also discusses a biomarker called 8-iso-PGF2 alpha, with which researchers use to measure oxidative damage in the body. Higher levels of this biomarker have been found in smokers and are associated with the increased risk of lung cancer. So tobacco damages cells in two different ways. It directly creates DNA adducts, but it also indirectly damages cells by producing oxidative stress, which increases the overall amount of cellular injury. Next slide. The last point I want to make is why cancer risk increases the longer someone smokes. Every cigarette introduces another round of carcinogens into the body. That means more DNA adducts are formed, more mutations develop, and DNA becomes increasingly unstable. Scientists call this genomic instability. As more mutations build up, more genes controlling cell growth become damaged. Eventually, enough of these protective systems fail that cells begin growing uncontrollably, which leads to tumor formation. This is why cancer risk increases both with the number of cigarettes smoked and the number of years someone has smoked. It's really the gradual accumulation of DNA damage over time that makes tobacco such a powerful cause of cancer. Overall, tobacco doesn't cause cancer through one single event. Instead, it repeatedly damages DNA, overwhelms the body's system repair, creates permanent mutations in important genes, and generates oxidative stress. Together, these molecular changes slowly transform a normal, healthy cell into a cancer cell. Next slide.
Okay. This section covers two connected questions. First, how many tobacco-related cancer cases and deaths are reported in the study? And second, which groups are most affected? I'm using Mansiga and colleagues' 2025 study in cancers, which examined U.S. adults, looked at incidence, mortality, and some subgroup patterns too. The main argument that they made is that the overall trend is improving, but the burden is still enormous and it's not evenly distributed. Before we get into the stats, it's important to understand what was counted in the study. They used United States cancer statistics data from 2001 to 2021 for incidence, and National Center for Health Statistics mortality data from 1975 to 2022 for mortality. Rates were reported per 1 million people and age adjusted to the 2000 US standard population. The cancers included, you can clearly see, not just lung cancer. A lot of people, there's a misconception when we talk about tobacco and cancer, they think it's exclusive to lung, but you can clearly see there's a long list of other tobacco-related cancers. And a limitation to the study to keep in mind is that they didn't look at individual smoking history. It was a comprehensive view of cancer burden for tobacco-related cancer sites. For the main statistics, though, the most important figures are the cumulative counts. From 2001 to 2021, the study found just over 14 million tobacco-related cancer cases among U.S. adults aged 20 and older. From 1975 to 2022, there were just under 15 million deaths caused by tobacco-related cancers. In 2022 alone, the death count was just under 350,000. The age breakdown shows why this is such a major public health burden for older adults. You can see the huge disparity compared to the other two age groups. So that is such a meaningful burden, and it really emphasizes why preventative measures are important early in life. Now we look at the 2021 incidence rates. The age gradient is pretty steep. The 20 to 49 had a rate of 453.1 cases per million. Adults 50 to 64 had a rate of just over 3,000 per million. And adults 65 and older had a rate of just over 8,500 per million. That means 65 plus incidence rate was almost 19 times higher than the 20 to 49 rate. Of course, this makes sense because cancer risk accumulates with age and duration of exposure. But like I said, prevention early in life is super, super important for that age group and in general. The distribution also changes by cancer site. In the 20 to 49 group in purple, colorectal cancer had the highest rate among tobacco-related cancers, followed by cervix and kidney. In the 50 to 64 group, lung and colorectal cancers were the two largest contributors, with lung slightly higher. In adults 65 and older, lung, trachea, and bronchus cancer is dominant at more than 2,800 cases per million, followed by colorectal cancer and bladder cancer. This is important because, like I said, everybody's thinking lung cancer and tobacco, but you can clearly see, especially in the older age groups, how, yes, lung is prevalent, but look at all the other cancers that are caused by tobacco. So for race and ethnicity, the key message is that most affected group depends on age. In 2021, non-Hispanic American Indian and Alaskan Native adults had the highest rates among younger adults and also middle-aged adults, 672.3 per million in the 20 to 49, and just under 4,000 per million in the 50 to 64 group. In the 65 plus group, non-Hispanic white adults had the highest rate at just under 9,000 per million. Non-Hispanic, Asian, or Pacific Islander adults had the lowest rates across all three age groups, and that disparity is one of the strongest in the entire study. Geographically, there are two important patterns. First, non-metro areas had higher incidence than metro areas in every age group. For example, 50 to 64, the non-metro rate was just over 3,500, while the metro had just under 3,000. Second, the region with the highest rate depends on age. South had the highest incidence rates for adults 20 to 49 and 50 to 64, while the Midwest had the highest rate in adults 65 and older. The West had the lowest rate across all age groups, but the study still found a small increasing trend among younger adults in the West. So, final takeaway is balanced. The study found that incidence and mortality have generally decreased in recent years, which suggests that tobacco control, screening, treatment, prevention efforts, all of those, they are having an impact. But the encouraging overall trends, they hide some important details and disparities. Among adults 20 to 49, incidence increase among non-Hispanic American Indian and Alaskan Native adults at 2.6% per year. Among Hispanic adults 20 to 49, incidence increased most recently from 2013 to 2021 at 1.7% per year. The West also had a small increase in young adults. So the conclusion is that tobacco-related cancer prevention still matters, but the step is targeted prevention, better access to cessation and preventative care, and attention to newer nicotine products such as e-cigarettes.
Now we will be covering what findings our group thought had the greater impact on the field. Next slide. For this section of our presentation, we wanted to focus on two of the most impactful and recent research findings related to tobacco and cancer. Both of these studies were published within the last 2 years in 2024 and 2025, and both appeared in highly respected peer-reviewed scientific journals. We specifically selected these papers because they examine two different but equally important aspects of how tobacco affects cancer. The first study focuses on the biological mechanisms behind tobacco-related cancer. Rather than simply showing that tobacco increases cancer risk, it investigates exactly how tobacco smoke damages DNA and contributes to the development and evolution of cancer cells. The second study looks at tobacco from a clinical perspective. Instead of focusing on how cancer develops, it examines what happens after someone has already been diagnosed with cancer. It asks whether quitting smoking after diagnosis can still improve survival, and the results show that it can. Together, these two studies provide a more complete understanding of tobacco's role in cancer. One explains the biological mechanisms that lead to cancer, while the other demonstrates how changing smoking behavior after diagnosis can improve patient outcomes. Next slide.
The first study was conducted by Torrens and colleagues and was published in Nature Genetics in 2025, which is one of the world's leading journals in genetics and cancer research. The researchers focused on head and neck cancers, which include cancers of the mouth, pharynx, and larynx. These cancers are strongly associated with tobacco use, making them an excellent model for studying tobacco-related DNA damage. To investigate this, the researchers analyzed the complete genomes of 265 tumor samples collected from patients across 8 different countries. Using whole genome sequencing allowed them to examine every mutation present within the cancer cells rather than looking at only a small portion of the genome. Their goal was to identify the specific mutation patterns left behind by tobacco exposure. They discovered 6 tobacco-associated mutational signatures, including several that had never been identified before. This finding suggests that tobacco smoke damages DNA through multiple biological mechanisms rather than a single pathway, making its effects much more complex than previously believed. Next slide. The major finding from this study is that tobacco does not just cause cancer through just one mutation or one type of DNA damage. Instead, tobacco smoke leaves several distinct mutational signatures throughout the genome. The researchers found that tobacco exposure was associated with higher overall mutation burdens, meaning smokers accumulated substantially more genetic mutations than non-smokers. They also identified differences in important cancer driver mutations. Driver mutations are changes in genes that actually promote cancer growth, allowing cancer cells to survive, divide, and spread. In addition, they observed copy number changes where sections of DNA are either duplicated or deleted. These genomic alterations can disrupt normal cellular function and further contribute to cancer progression. Interestingly, the study also found that different parts of the head and neck accumulated different mutation patterns. For example, cancers of the oral cavity showed different tobacco-related signatures than cancers of the larynx. This suggests that different tissues respond differently to tobacco exposure. Overall, the researchers concluded that tobacco doesn't simply initiate cancer. It continues influencing how cancer cells evolve throughout the progression of the disease by creating multiple forms of genetic damage. Next slide. We chose this study because it provides some of the strongest molecular evidence available showing exactly how tobacco contributes to cancer development. For many years, researchers have known that smoking causes cancer, but this study moves beyond that general statement. Instead, it identifies the actual genetic fingerprints that tobacco leaves inside tumor DNA. This is significant because these mutational signatures allow scientists to trace the biological history of tobacco exposure within cancer cells. Understanding these mutation patterns may eventually help researchers improve cancer prevention strategies, identify individuals at higher risk, and potentially develop more personalized treatment approaches based on the genetic characteristics of tobacco-related cancers. Another reason we chose this paper is that it represents how cancer research is evolving. Modern cancer research is no longer focused only on identifying risk factors. Researchers are now trying to understand the precise molecular mechanisms that drive disease, and this paper makes an important contribution to that goal. Next slide. The second study was conducted by Sincirapini and colleagues and was published in JAMA Oncology in 2024. Unlike the first paper, which focused on cancer biology, this study examined a very practical clinical question. Does quitting smoking after a cancer diagnosis actually improve survival? The researchers studied 4,526 patients who had already been diagnosed with cancer and who were current smokers when entering treatment. All patients participated in a structured smoking cessation program provided through MD Anderson Cancer Center. This program included individualized counseling sessions, medications such as nicotine replacement treatment and therapy, or other pharmacological treatments. And because most visits occurred remotely, over 95% of patients received their treatment through telemedicine. The researchers then followed these patients over time and compared survival outcomes between those who successfully quit smoking and those who continued smoking. Next slide. The results were very clear. Patients who successfully quit smoking consistently had better survival outcomes than patients who continued smoking. Improvements in survival were observed among patients who remained smoke-free at 3 months, 6 months, and 9 months after beginning treatment. Perhaps the most important finding was that timing mattered. Patients who entered smoking cessation treatment within 6 months of their cancer diagnosis experienced the greatest survival benefit. For this group, survival at the 75th percentile increased from approximately 2.1 years for patients who continued smoking to approximately 3.9 years for patients who quit smoking. This study also found that quitting later still provided benefits. Although their survival advantage gradually decreased as more time passed after diagnosis, this suggests that smoking cessation should begin as early as possible during cancer treatment. Next slide. We believe this study has enormous clinical importance because it changes the way we think about smoking cessation. Traditionally, quitting smoking has been viewed mainly as a way to prevent cancer before it develops. However, this study demonstrates that quitting smoking remains beneficial even after someone has already been diagnosed with cancer. The findings suggest that smoking cessation should be considered an essential part of cancer treatment rather than simply a lifestyle recommendation. By helping patients quit smoking early, healthcare providers may improve treatment effectiveness, reduce complications, decrease the risk of additional cancers, and most importantly, improve overall survival. Because these findings have direct implications for patient care, oncology clinics may increasingly incorporate smoking cessation programs into standard treatment plans. This makes the study highly relevant, not only to researchers, but also to physicians, nurses, and patients. Next slide. When we look at these two studies together, they provide a complete picture of tobacco's impact on cancer. The first study explains the biological mechanism by showing that tobacco leaves distinct genetic fingerprints in DNA, causes multiple mutation patterns, and influences how cancer cells evolve over time. The second study demonstrates the clinical consequences by showing that quitting smoking after diagnosis significantly improves patient survival, especially when cessation begins early. Together, these studies highlight that tobacco affects cancer at every stage, from initiating DNA damage that contributes to cancer formation to influencing patient outcomes after diagnosis. This is why we selected these two papers as the most impactful recent findings. One advances our understanding of cancer biology, while the other directly informs patient care and clinical practice. The overall message we want everyone to remember is that tobacco control is important throughout the entire cancer journey. Preventing tobacco exposure reduces the risk of developing cancer, and for patients who already have cancer, quitting smoking as early as possible can still significantly improve survival and long-term outcomes. Next slide.
All right, so now we're transitioning over to the conclusion, and here we're just going to reiterate everything we've already discussed in the presentation, but also prevention, quitting, screening, and future research, and why all of this is important. Next slide. So what have we learned? So in conclusion, tobacco continues to be one of the leading preventable causes of cancer throughout the world. We discovered during this presentation that tobacco smoke contains thousands of dangerous compounds, including over 70 carcinogens that cause genetic mutations and, of course, damaged DNA. These alterations eventually cause cells to grow uncontrolled and turn into cancer. We also discussed how tobacco can affect areas other than the lungs. So because toxic chemicals enter the bloodstream after being smoked, smoking has been connected to cancers that affect numerous organs throughout the body that were mentioned throughout the presentation. So this explains why a sizable portion of cancer cases and fatalities each year are still considered to be directly tied to tobacco use. So how can cancer be prevented? Another important point from today's presentation is that prevention can be considerably more successful than just getting treatment on its own. But even for those who currently smoke, giving up while you still can have a significant effect on your health. According to one of the studies we covered, people who quit after being diagnosed with cancer fared much better in terms of survival than those who kept smoking anyways. So this proves that quitting is never too late and is always still an option. But why is early detection important? Early detection is also an important part of reducing cancer deaths because individuals who have a long history of smoking and are therefore at risk should follow recommended screening protocols, and particularly if they have lung cancer. Early cancer detection offers patients more treatment options and significantly increases their chances of survival. But what's next? In the future, scientists will probably continue to investigate how tobacco harms DNA and how these genetic alterations impact the development of cancer. New discoveries may lead to improved screening methods, more personalized treatments that are based on patient-by-patient cases, and better smoking quitting programs that help more people quit successfully. All things considered, it seems pretty clear. Tobacco use is a significant contributor to cancer that can be prevented by either quitting smoking altogether, earlier detection, educating the patients and people in general, and general prevention that can save many lives. We can lessen the weight of tobacco-related cancer in the future by promoting healthy choices and carrying out more research on the topic. But that's basically it. Thank you so much for your time. I'll go over to the reference slide. And these are all the reference that we used and referenced in and throughout our presentation. Next slide.
Thank you.
Thank you.